Hello. Here we go. Welcome to our second Ecology Center seminar speaker of the year. It's my pleasure to introduce Priyanga Amarasekare. Been practicing that. Priyanga grew up in Sri Lanka. I like to ask our speakers, when was it in their career that they realized they wanted to become an ecologist? She told me it wasn't until she scored 1 point too low on the qualifying exams in both law and medicine. She couldn't become a lawyer or a doctor, ended up in bioscience, and then had a class on diversity that really got her excited about the field, and she never looked back from there. So we can thank serendipity for her contributions to ecology. She did a master's at University of Hawaii, which she completed in 1991. She worked on nest predation. She then went to UC Irvine to work with Steve Franks, completed her PhD there. She worked on host-parasite interactions, started taking a lot more math classes, developed her theory chops, and that's also where she started working on spatial heterogeneity. I'll come back to that in a sec. Went to do a postdoc at NC's in Santa Barbara, then joined the faculty at the University of Chicago, and then moved to UCLA. Her work focuses on patterns of biodiversity, on how spatial heterogeneity promotes coexistence, how that's fueled as well by species dispersal. She also looks at the impacts of climate change on biodiversity, looking at specifically at how really metabolic rates influence consumer resource interactions. She's highly decorated. I'm not sure I've ever introduced a speaker who's won as many of the big awards that are available in our field. She's won the Young Investigator Prize from the Society of American Naturalists. She was elected a fellow of the Ecological Society of America. In 2021, she won a Guggenheim Fellowship. And just this last year, she was recognized by the Ecological Society of America with the MacArthur Award. I think it's the most prestigious award that the ESA gives. I do want to recognize, though, that her recent career hasn't been all about awards. So many of you may have read articles in Nature or in the Chronicle of Higher Education about the terrible treatment that she's received over the last couple of years from UCLA. It's included a 1-year suspension without salary, without benefits. I think it's remarkable that she's continued to do science in the face of that kind of adversity. And in fact, this morning she told me that one of the things that helped her survive that time was being able to focus on her work for perhaps a few hours a day and finding a little peace in the work itself. I know that our grad student committee that invited Priyanga thought that this invitation would be a way of showing our solidarity with you. And so we're really grateful that you accepted the invitation. Welcome.
Thank you. You can hear me? So one thing, I never wanted to be a doctor. I did want to be a lawyer. I miss both. And it's a great pleasure to be here. And I want to thank Mitch, Annie, and the Graduate Student Committee for inviting me. It's been wonderful being able to talk to students, and I really appreciate it. Being able to get away actually really helps me also because it connects me with science. And the other thing I want to mention is the two particular highlights I hadn't expected: the maples and the crickets on the sidewalk, which I found very entertaining. So, so tomorrow I'm going to give a seminar that's more focused on my research. And so today I want to talk about something a little more general about how we do research, teaching, and mentoring in this rapidly changing world with increasing awareness of racial and social justice and the need to connect science with history and society. So now this is really a topic for a discussion rather than a formal seminar. So what I thought was perhaps I could give the research, cover the research component, which is fairly brief. And if you like, we could have a discussion at that point. And I really think the teaching mentoring part should be a little bit more interactive. And I'd like for you to ask questions if you like, if you want to. Okay, right. So let me start with the research component. So in 2019, the National Science Foundation sponsored a working group on advancing theory in ecology. And a number of us was invited to this working group and were asked to come up with priorities. And I led a subgroup focused on integrating theory across levels of organization along with Roger Nisbet. And I want to present some ideas we came up with in our particular subgroup, which is eventually going to become an article in a special feature in ecology. So now, understanding how lower level processes translate into higher level outcomes is a fundamental challenge in ecology. It's also a key priority for addressing the environmental problems that plague the planet. Now, our subgroup identified 3 priorities for advancing theory to address this question. The first is to investigate mechanisms by which suborganismal processes, so processes that occur within organisms, translate into population and community level processes. The second is to investigate cross-level interactions between individuals of one species and populations or communities of others. The third is to investigate conflicts, commonalities, and robustness across levels of organization. And our goals were to propose new directions for theory and to inspire new directions for empirical research. And here I have outlined some of the 3 topics and some of the top subtopics that I'm going to touch on today. I'm not going to go through everything we came up with for interest of time, but I chose 3 that I think might be of broad interest to this audience. So let me start with the first subtopic of the first topic, which is predicting species responses to environmental perturbations. We focused on climate warming, not least because it is the defining environmental crisis of our time. And we identified 3 priorities for addressing this issue, which I am going to discuss in turn. Now, predicting the effects of climate warming requires understanding how temperature effects on phenotypic traits translate into the dynamics of populations and communities. It's essential that we make this connection if we are to make predictions. Now, one important priority in this regard is to develop a common mechanistic framework for characterizing temperature response functions at the phenotypic level. Now, the typical approach is to use reaction rate theory to characterize temperature response functions. These characterizations are either monotonic or left skewed. But thermal reaction norms of reproductive and consumptive traits do not fit this mode. They tend to be symmetrically unimodal, as you can see from these data here. So these are the birth rates, reproductive rates of a number of different species of arthropods. These are attack rates and maximum uptake rates for a large number of taxa from spanning all groups of invertebrates up to reptiles. So you can see that these responses are more uniformly less skewed, so unimodally and more Gaussian or symmetrically unimodal compared to the left skewed response I showed you before. So what this mismatch then illustrates, the reason for this mismatch is that reaction rate theory characterizes what are called rate control processes. So these are reactions that proceed to their maxima, followed by a rapid decline at high temperatures when there are things such as enzyme denaturation, protein unfolding, and things like irreversible changes happen. Now, reproductive and consumption traits are regulated by negative feedback processes driven by things like neural and hormonal regulation. As a result, these reactions are prevented from increasing to their maxima. They actually are regulated so that there's a feedback that drives towards an intermediate optimum. So what we need now is given how important these traits are for, for fitnesses of organisms, we need a framework that integrates the temperature dependence of these regulatory and rate controlled responses. I'm going to talk a little bit more about this tomorrow at my seminar. Now, the second priority is to incorporate the epigenetic changes underlying short-term responses such as acclimation into ecological models. Now, phenotypic responses to environmental perturbations involve epigenetic changes such as DNA methylation and histone modification, which actually do not alter the genetic code. They're not in the DNA sequence that determines your genetic code, but is extra to that. So ectotherm development provides one of the best examples of epigenetically driven phenotypic responses. So these are things we've already known about for a very long time. So acclimation, then, the short-term response to an environmental perturbation, results from epigenetically driven gene expression changes. A key component of the acclimation response is the time delay between when cells sense a change in the environment and activating the cellular machinery involved in gene expression and protein synthesis in response to that environmental change. So there is a delay that we need to take into account. So making accurate predictions of short-term responses to warming if you want to make accurate predictions of short-term responses to warming, it's critical that we incorporate this cellular level delay into ecological models of thermal acclimation. So the point is, it's not an instantaneous response. There's some time delay, and that delay may make all the difference between the stimulus and the response. Now, this is particularly important because epigenetic changes can be transmitted from parent to offspring across several generations, especially in plants. It's really critical in plants. So that could potentially lead to genetic assimilation via mutations that incorporate these epigenetic changes into the genetic code. So really getting this information into our ecological models is very, very important, especially since warming is the kind of process where we need to take into account both ecological and evolutionary responses. All right, so I've talked about two of the priorities under this very first topic. And the third one is about reaction norm evolution in non-stationary thermal environments. So I want to explain a few things here. A reaction norm is the range of phenotypes expressed by a given genotype in response to environmental variation. Non-stationary means warming, as you know, is a thing that keeps changing, right? So what it means kind of formally is that the mean and variance of the process keeps changing. So, like seasonal variation is a form of variability, but it has a certain predictability. We know that it comes up and down, and we could do that. Warming is not like that. It keeps changing over time. So it's formally we say it's non-stationary. I just want to demystify the jargon there. Okay, so there is a body of theory on reaction norm evolution in temporally variable environments developed mostly by Russell Landy. But this theory considers linear reaction norms, largely for reasons of mathematical tractability. But we know that thermal reaction norms are highly nonlinear. And we can't really, so for example, it used to be that you would just take this linear portion and say that approximates the actual response. But we know that there's mortality keeps increasing, but also, look, these things actually decline at high temperatures. So we can't ignore that nonlinearity anymore and assume that it's linear. So theory that can a key research priority for the future is theory that can incorporate the evolution of nonlinear thermal reaction norms in non-stationary thermal environments. And we also need a mathematical framework that can incorporate reaction norm evolution in non-stationary environments into models of ecological dynamics. Okay, so I've talked about this one particular subtopic and our first main topic. The second one, now I move on to the second one, cross-level interactions. And I'm going to talk about this in the context of host-pathogen interactions. Now, negative frequency-dependent selection and niche partitioning are mechanisms that maintain diversity at different levels of organization. They share a common underlying mechanism. Entities, be they genes or species, experience few or no negative interactions when rare, and mostly self-limitation when abundant, intraspecific competition. Now, let's consider this in the context of a particular life history. Long-lived hosts and short-lived pathogens or parasites. Trees and seed predators, human and wildlife diseases provide good examples. So what this life history difference implies is different diversity generating mechanisms in hosts and pathogens. Because they're short-lived, pathogens can accumulate genetic diversity on ecological timescale. But because they're long-lived, intraspecific variation in the host is most likely non-genetic on that timescale. Think about trees, or even like elephants, or anything long, any mammal, for example, compared to the lifetime of a pathogen. So why does that matter? It matters because having an advantage when rare necessarily means being disadvantaged when abundant. Rare types do not experience negative biotic interactions, and they can increase when rare, provided the abiotic environment is not unfavorable. The challenge is how to persist when abundant. So I'm going to discuss this using an example of work done by Mercedes Pascual and her colleagues on the malaria pathogen. Now, Plasmodium exhibits high genetic diversity generated by mutation and recombination and maintained via negative frequency dependent selection and niche partitioning. So pathogen strains have an advantage when rare, obviously because hosts lack immunity to new variants, right? We all know that. Strains have a disadvantage when abundant because now hosts develop immunity to common types. But if hosts differ in their specificity such that each host is immune to only a few pathogen strains, then the common pathogen strains can partition host types such that they experience stronger interstrain competition compared to interstrain competition. Thus, niche partitioning allows common strains to persist over epidemiological timescales. Now, the operative word there is epidemiological timescales, which, as we know, are relatively short. And the key thing for the pathogen is to persist over the epidemiological timescale. Right. So this provides this idea, and this example provides a general mechanism for antagonistic interactions with long-lived resource species and short-lived consumer species, where cross-scale diversity is generated by different mechanisms. It holds the promise for a general theoretical framework for consumer resource interactions with different life histories and different sources of intra and interspecific variation across trophic levels. Oops, what did we just do? All right, so that's topic number 2. Let me talk about third topic, conflicts and robustness. And I will start with conflicts across levels of organization. Now, Public goods and common pool resource problems are central in ecology. They range from tumors to nitrogen fixation to vaccination and mask wearing. These problems are essential to understanding the emergence of structure and pattern in ecosystems, investigating comparable problems in economics and social sciences, and for tackling environmental challenges. To address these things, we need theories that can incorporate scaling from individual to system level, emergence of prosociality, collective decision-making, and cooperative graves. Now, these are ideas you might think, well, we've already got established ideas. These are established ideas for which there exists a large body of theory. But the thing is that new problems keep cropping up, requiring new innovations in the theory. For example, vaccination and mask wearing represent cases where individual freedom based on religious and political beliefs is pitted versus the common good. Self-destructive behaviors like my body, no mask, provide the raw material for selection. So you can see that these ideas of, the whole idea, going back from the tragedy of the commons, right? Sort of basically self-interest versus the good of the community. They keep cropping up time and again. Okay, so that's about conflicts. And now let me talk about the second topic, robustness across levels of organization. And I'm going to do this in the context of network topology and energy flow. So networks of interacting entities occur at all levels of biological organization. We have gene regulatory, transcription, and neural networks at the individual suborganismal level. We have social networks at the individual level, and we have species interaction networks at the community level. Now, ecological communities are both similar to and different from suborganismal networks. And we happen to know a great deal about suborganismal networks. So similarity arises from the modular structure, where a module is a subset of entities, be they genes, cells, or species, that interact strongly amongst themselves and only weakly with other entities or subsets. Ecological communities differ from suborganismal networks in that suborganismal networks tend to transfer information, whereas ecological networks tend to transfer energy. So there is this existing body of theory on suborganismal networks, which shows that modularity, this division, divisible into subunits that interact strongly within themselves but weakly with others, can both buffer perturbations that occur in a community as well as maximize information transfer. This suggests two priorities, two new directions for ecological theory. The first is the robustness of ecological networks to perturbations. Can they still stay cohesive, right? Can they still be unchanged, relatively unchanged when subject to perturbations? Now, we know a great deal about biotic perturbations such as species additions and removal, and how those things affect things like network topology and community dynamics. Network topology just simply means if you draw out a food web, that's a topology, right? That's it's a pyramid, it's a web, it's, whatever that is, that's a topology. And the dynamics are, of course, how these things change over time. We happen to know relatively little about how abiotic perturbations such as heat waves and toxins influence these properties of topology and community dynamics. So theory that investigates the effect of abiotic perturbations on network topology and community dynamics is therefore a key research priority, especially given the pervasive problems of pollution, climate warming, inflow of toxins, etc. Right. Now, the second direction is about determining what sorts of network topologies maximize energy transfer. It's a really interesting question. What is it? Is it just a chain? Is it a web? Is it a triangle, a diamond? What maximizes energy transfer? Because you would expect that modules that maximize energy transfer actually do better overall than modules that do not. So we could hypothesize that we see the modules we do see in nature because they tend to maximize energy transfer. So, but how do we test that? The one problem with the existing theory is it's framed in terms of species abundances. But in order to actually look at energy transfer, we have to convert these things into units of carbon, units of energy. So we need a different state variables, different conversions. We need new theory that can integrate the stoichiometry of nutrient and energy transfer with the dynamics of species interactions. And we need such a framework can help us identify the community models that are robust to perturbations and can also maximize energy transfer. So with that, just to summarize, we have this subgroup tasked with coming up, or like a focus that focused on integrating theory across levels of organization, from cells to organisms to populations to communities. And we've identified 3 priorities for advancing theory that's actually at the interface, even though I said ecology, at the interface of ecology and evolution. And I've shown you, I've discussed a few topics under each of these things. So with that, I want to thank the National Science Foundation actually for sponsoring this workshop and bringing a lot of us together and inspiring us to come up with these ideas. And at this point, I would like to stop and see whether you have any questions. Okay, I will continue, and then maybe you can ask me questions later. And I know it's a sort of a lot of different topics. I did want to talk about that rather than anything else, because I thought it was such a new thing that we've done. Maybe this would like make you think, oh, there is a question.
Could you just talk a little bit more about the ecological systems try to transfer energy, whereas evolutionary or cellular ones transfer information? And you talked about those systems that are most efficient at transferring energy, maybe being more stable. Could you talk a bit more about the kind of mechanisms behind that? In evolution, we have selection that where genotypes or phenotypes are the ones that are most fit are propagated. So there's that kind of structure to keep those that are most efficient at transferring information. But could you talk a bit more about that?
So it's interesting. So, if you think about natural selection, that operates at the level of an individual. So anything that increases the fitness of an individual will, of course, provided it's heritable, will then spread, right? So any kind of, let's say, transcription network, a neural network within an organism that enhances its fitness is going to have more copies of itself made. Now, when you come to ecological communities, we do not have heritability in that same sense, right? We don't have transmission. What we do have is a sort of a thing where we would expect certain configurations to persist and certain configurations to fail. That's simply that's like startups or anything, right? So you can have selection. What we don't have at that level is heritability. You certainly can have selection. So the idea is, it's just like public goods, right? So it's a different question, but it's the same thing. It's self-interest versus the common good. If only you could put the common good above the self-interest, everybody would do better. But often that doesn't work unless you're related to your neighbors. So in the same way, so the idea is that it's like a game. I think of it like actually game theory. So there is going to be an uninvasive configuration in terms of this energy transfer, which I'm actually testing that sort of idea. So let's say, you look at actual food webs, let's say food webs, right? Food web configuration. So we could have diamonds, we could have trophic chains, we could have triangles, inverted triangles. so the question is, which of those are least susceptible to invasions? Because if it can be broken and a link can be broken and a different link could be made, that means there's something sort of, better at, so basically that's not stable to a perturbation. So the whole idea of modularity is that they're robust to perturbation. So there's got to be, so my way of thinking, I haven't actually, this is sort of new, like, this is something we're starting out. I don't have everything worked out, but that's my idea. There's going to be an unbeatable combination, so to speak, within the constraints of organisms being able to get energy, to use energy, and to persist in the phase. And then when we have environmental perturbations like what Communitech's facing now, a lot of this it's in a different realm, right? Those unbeatable, that how we think about those things now have to change with respect to the kinds of perturbations that are being imposed. So example, a community that's robust to warming may not be robust to toxins because it's a very different sort of aspect. So there's a lot, but I think that if we understand what sorts of configurations maximize energy transfer, we might understand why we see certain configurations to be more stable in these systems. The whole network topology idea is there are recurrent motifs. There are repeated patterns, right? So why do certain patterns repeat themselves but not others? And I think the key to understanding that is this sort of idea, because, being inspired by suborganism, did I just lose my, no, suborganismal network theory, the ones that we see persistent are the neural networks that are most efficient at information transfer. So that's the idea. Okay? Yes.
Part where you're talking about the niche partition
Yes.
of the host versus the pathogen. Can you go into why that works with the common strain while the niche partition?
So it's that idea. So it's where, so you have pathogen strains that are now common. The problem with being common is most hosts are resistant. So then you're going to go extinct, right? But the idea is that the hosts themselves are heterogeneous in their ability to resist pathogen strains. So basically it's saying hosts are only specialized, their immune system. So you can actually think of it as, you can either have a generalized immune system that resists everything, but is not very good at resisting any particular strain. So you're ill halfway ill the whole time, or you're very, very good at resisting particular strains, but not others. And the chance of, it's a different strategy. So this is actually talking about the latter strategy. And this is now, I have to say, that's inspired by, that's observed in the malaria pathogen and that system. Whether that actually works in a lot of other systems is open to question, but it certainly does work in, I think, the whole Janssen-Connell idea. The pathogen, seedling predation. It's another example of that sort of thing. So the idea is that if hosts have a constraint on how much immunity it can develop against the strains, so say host A is resistant to 4 strains, host B is resistant to another 4 strains. These 4 strains can then attack host A. And so it's niche pathogen. But actually, if you really let these things run, it's going to go where something wins. It's just, but that's the thing. It's epidemiological timescales are very small. We know it's transient, right? An epidemic comes and goes. All that matters for the pathogen is to be able to persistent on the timescale at which it can transmit itself to another host. So that's how it works. So it's a niche partitioning of the pathogens of hosts. So each host is a niche for a set of strains of pathogens. Anything else? Perhaps I should get to the next part and then we could have a longer discussion at the end. Okay. So, and I think these are questions that you might actually want to think about more, and I'll be happy to take, answer more questions tomorrow. Okay. So now I want to get to the second part about teaching and mentoring in this rapidly changing world. And I want to talk about how my own philosophy towards teaching and mentoring has changed in response to this sort of greater awareness of racial and social justice, and the need to connect science with history and society. And for this part, I really think if you want to ask me a question, please do, because I am grappling with these things myself. I don't really have answers, really. I just know what I try to do. And I'm always looking for advice or guidance and discussion, about these things, because it's new to me as well. So my sort of primary goal in teaching is to teach students to use quantitative reasoning to understand the complexity of biology. Now, I show them that complexity arises from underlying nonlinearities, and that intuition is a poor guide for understanding nonlinear phenomena. So that then paves the way, when you sort of make them understand that, it paves the way for introducing the need for mathematical approaches. So that's what I try to do. Now, there are a number of challenges associated with taking this approach. One thing is that most biology students have a fear of mathematics, which goes back to bad experiences in calculus. I mean, I'm sure we all have traumatic experiences of doing calculus within 50 minutes and not getting it right. But what the thing you have to understand is when you're a scientist working with it, you're not doing a 50-minute exam. You're sitting down with a stack of paper and you're working this out. And nobody's like looking overhead to give you a grade. And you'll be amazed at how differently you think about it, right? So that's really like the thing that I try to tell students is that this is not a test. And it's just that once you actually know the context for why you need to take a derivative or why the Taylor series is so important, it becomes such a different, you look at it so very differently. Anyway, and the second challenge is a different one. It's the sort of the racial, ethnic, and socioeconomic disparities amongst, between students that take these courses. The idea that Black, Brown, Indigenous people are inherently inferior in mathematical and quantitative ability is an idea deeply ingrained in this society. And socioeconomically, students from socioeconomically disadvantaged backgrounds have additional hurdles due to unequal access to resources, regardless of their race, gender, or ethnicity. So these are things that sometimes we are not aware of, that they're struggling with these issues, and that we need to be, as teachers and mentors, be aware of and pay attention to. So over the years, I've tried to develop a teaching program that can accommodate these disparities and negative perceptions. And I didn't do this when I started out. I've learned to do this and I'm still learning to do it. But I teach teaching. I sort of regard it as an exercise in adaptive management, meaning that you learn and correct at each stage. So my approach. So I motivate students by introducing questions with societal implications and illustrating the need for models. This is very important if you're at a place like UCLA, where the majority, 90% of the students are pre-meds and have no interest in ecology. But the minute you mention a cancer or disease, then they sort of perk up and start paying attention. So there's a sort of a way to get people interested. You just have to be able to kind of find a way to resonate with them. And I also empower students by example, and I just talked to them about my own background as a biologist with no formal training in mathematics, and sort of, teaching myself to, learn these things, which is, which, generally sort of allays at least some of their fears. So I demystify models to show that pretty much anyone can learn the mathematical quantitative methods need to study ecology and evolution. For example, I derived the logistic equation from scratch to show that the density-dependent function is just the equation for the straight line. It's actually y equals mx plus c. And once you show that, it's so interesting how much more comfortable students feel and how much more receptive they are to the slightly more complicated models that are to follow the logistic. But it's a way to show that it's nothing different from what they know. And it's really important. So at least in my case, because I do teach somewhat quantitative courses. And I try in my courses to connect science with history and society. And lately, in the last several years, I've tried harder to do this. And these are two examples that I've come up with for this talk. So I think it's very important that we learn from history so that we can refute the false arguments that are continually being made in the socio-political arena, and to prevent future misuse of science to harm disadvantaged minorities. And you see and become, as more and more, when you see this in the public, sort of the arena, the more the responsibility that I personally feel. So one example I want to use is a course in epidemiology that focuses on the intersection between history of epidemiology and the lived experiences of Black people. So you can teach the conceptual basis of disease spread using classical models and deriving or not the basic reproductive number, which is what most of us do in classes. But you can also give the historical context of disease spread in terms of the slave trade and the spread of yellow fever in the Americas and the decimation of indigenous peoples through the introduction of diseases such as smallpox. You can teach the conceptual, you can use epidemiological models to teach the conceptual basis of public health measures such as vaccination and herd immunity. Again, something we routinely do. We can also talk about Black people's mistrust of public health measures, having its roots in historical events such as the Tuskegee study on syphilis, forced sterilization of Black and indigenous people, and the view actually prevalent amongst a lot of Black people that AIDS epidemic was created to harm them. So these ideas, this historical backdrop provides the backdrop for present-day skepticism of vaccines, for example. We can introduce students to the poverty diseases, such as malaria that motivated the first epidemiological models, tuberculosis and AIDS, which account for 40% of the disease burden in some of the poorest nations in the world. In the same way, we can connect climate warming and human health by introducing scientific studies that highlight socioeconomic disparities. So there are studies now showing that warming reduces the human gestation period and increases the number of stillbirths on extreme hot days. These problems disproportionately affect Black people and other disadvantaged minorities. I'm sure a lot of you listen to NPR. NPR has this program called Our Body Politic that deals with these kinds of issues and other issues as well, but they actually deal with a lot of these socioeconomic issues. So that's my first, wait, did I get this right? Yes. So that's my first example. And my second example is a course on conceptual foundations of ecology and evolution, which a lot of us actually, teach and learn. And I think like a course like this, I believe that a course like this should emphasize how underlying biological differences have been used to support bias and prejudice against particular groups such as women, African Americans, and Latinos, and indigenous people. This brings us right into the thorny topic of eugenics and its role in shaping world history, from slavery to the Holocaust, to genocide of indigenous peoples and modern day ethnic cleansing. So the question is, how does one teach conceptual foundations of ecology and evolution against this backdrop? How much responsibility do we have to educate the young generation about this history? So I've had to grapple with this myself in this recent course I developed on, which I call the imprint of racism and sexism in ecology and evolutionary biology. So I've structured this course around 4 themes. I'm not going to go into all of them. I want to just highlight a couple for this talk. So Darwin's ideas of natural selection was inspired by Robert Malthus's essay on principle of population. Now, Malthus, in turn, was inspired by the idea that if one could only get rid of poor people, it would be easier to achieve population regulation. This is a lesser known fact about Malthus. He used to attend the church in his village, and he was appalled by all the poor people who came. Well, let's get rid of them. We have population regulation. And that's something we don't know about. We all know about his essay. We don't know about why he was motivated. Now, a key aspect of the modern synthesis is the integration of Darwin's theory of natural selection with Mendel's laws of inheritance. But this synthesis also brought to the fore ideas and practice of eugenics. So, R.A. Fisher. R.A. Fisher was a brilliant man. He founded the field of population genetics along with J.B.S. Haldane and Sewell Wright. He was the founder of modern day statistics. Anything you do in statistics, you will check on it. Fisher started it. Fisher developed them. He developed the first mathematical model of Mendelian genetics and natural selection. But Fisher was also a prominent eugenicist who believed in white superiority and supported Nazi eugenicists even after World War II. That's something we generally don't teach, but I've actually started to talk about, and many people now in conferences qualify their talks, research about Fisher with his history, which I think we should all be aware of. It's a question of, I think this happens with artists too. Do you separate the science from the person? How do you actually reconcile these different parts of people who have made contributions? So the rise of eugenics in the United States was actually, it began after the discovery of Mendel's laws and the possibility of breeding for specific traits. This gave rise to scientific racism and the perceived inferiority of Black and Indigenous people. And it led to shameful practices such as forced sterilization of poor and disabled people and Black, Native American, and Latin women. So teaching the principles of natural selection and population genetics in this historical context also provides a conceptual foundation for modern day issues of diversity and inclusion and the ethics of medical genetics. I don't know how many, we read a few papers on this about like these studies. You do these research studies about the efficacy of drugs, treatments. Minorities are very underrepresented in these studies. So, they only apply generally to people of European descent, and whether those things actually could work with other groups are typically not even considered in these studies. In the same way, we could talk about the ecological foundations of environmental catastrophes in the context of the disproportionate effects of climate change on indigenous people, countries in the global south, and non-white areas of many US cities. We can and in the environmental management practices in the context of Black, Brown, and Indigenous people's fight for environmental justice. This study came out in Nature last year, and it shows that warming is going to increase the risk of floods, and this risk will disproportionately affect Black communities, especially in the South. All right, so that's my philosophy on teaching. I don't know, how am I doing for time? 9 minutes? Okay, let's see. All right, so my approach to mentoring is also shaped by my personal experiences as a person with multiple overlapping identities. I'm an immigrant from the global south. I'm a woman of color. I work in a field, theoretical ecology and evolution, where women are underrepresented and women of color are very rare. I am also in this unusual position. I hadn't even realized this for a very long time, being both the donor and recipient of diversity efforts, and the responsibility that places on me. So these experiences and insights have allowed me to develop mentoring strategies that help minorities navigate these challenges and also to educate non-minorities in issues of EDI. So my goal with graduate mentoring is to train students to take an integrative and quantitative approach to research while helping minority students navigate the challenges of racism and discrimination, and also providing a safe environment in which they can speak and act without fear of reprisal. It could be something really simple, like asking, what does that mean? Like a question, because a lot of people think, well, I don't know this, but probably everybody else does. And then you never ask the question because you don't feel comfortable. You went to a school where they didn't teach these things. You didn't have computers. You didn't get a laptop to take home. So, there are things that you don't know about these students that sometimes really impede their progress. And I also try to empower by example. I talk about my, own attempts to learn mathematics on my own and, achieving some measure of success even in the face of challenges, and which sort of, helps people to know that you've also had faced challenges and you'd managed to overcome them to some extent. So with students, I actually kind of take this very long-term strategy. So I tell them we need to devise some sort of strategic plan, set up long-term goals, and a time frame for achieving these goals. Believe it or not, I do that with every single student of mine. Like, what do you want to be after you? You want to get a postdoc and a job? Here's the plan. Let's try to do this. Year 3, this, year 4, this, year 5, you're out of here. So it doesn't always work, but mostly it has actually worked. So we get an early, I give the students, I tell the students, I facilitate their getting an early start in developing quantitative skills, which is critical for confidence building in female and minority students. And I encourage all students to have a component of theory in their thesis. It's not required, but I encourage them to do that. And we plan ahead so that the students can, finish their coursework and get their publications ready so that they're ready for can move on to their next career phase, hopefully by year 4. And I also try to help students shine in their own light. So I encourage them to pursue their own interests. So I always say, I want you to be known by what you do, not by who you worked with. Right? So I want them to try to do their own thing. So do something a little bit different from what I do, so you could take it with you. And that's how you're going to be known one day. So I want to just take a few minutes. How many more minutes? I want to talk about undergraduate mentoring. This is my absolute passion, my proudest achievement. I really believe that early training is the key to producing those exceptional future scientists. And, you might just turn some of those pre-meds who really have no idea why they're in pre-med to find out that they love ecology, or they actually really are happy doing something else. Not that I really am changing the minds of pre-meds, but I have had students who did change track. And so what I do is, it's a bit unusual. I personally supervise these students. I help if they sort of survive for 2 weeks. I say, here's my lab, here's what you do. You have to come every day or every other day, but you'll get these really interesting results. And if they survive the 2-week challenge, I sit down with them, we develop independent projects. I actually supervise them. And so with this, it's a lot of work, but it's been really rewarding. And I've got the highest, I'm actually going to brag about this, I've got the highest number of honors students. I've got the highest number of awards, winners, fellowship winners. And my students have first authored papers in AMNAT, JE, and it's just really been, even the ones who went to, so one person who got a paper, well, the person who got AMNAT went to Chicago for grad school. One person who got JE still wanted to be a doctor, but he went to Johns Hopkins. And he did take a year off to do a master's in public health. He said, well, you always tease me about not doing research. Yeah, I'm doing research. Well, anyway, he's a very good doctor. But I'm not sure I take credit for that. But anyway, so the reason I brought this up was also wanted to say first for graduate students, when I was a graduate student, I actually had undergrads. They technically signed up with my advisor. He was a theoretician. He didn't do experiments, but they helped me with my research, and they presented in symposia. They won prizes. It was actually very rewarding and gratifying. I did it just because I thought this is fun. Let's go out in the field, let's do some things. I was learning about these insects. But then it also had an unexpected perk. When I first became an assistant professor and I had to write a grant for NSF, I already had broader impacts. I'd already mentored students. So you see, like, it is actually, if you think of like plan your career ahead, and I would say if you're a graduate student doing field work or you're a theoretician, whatever you're doing, try to engage undergraduates. It's really like when you find the right people and to see them get so excited, because they're young and this is just like you just like can make immense impact on people's lives. So with that, I'm going to stop, and thank you very much for listening.
Okay, before people run away, I forgot to remind you in the intro, there is a social tonight at Karen Mock's house. Here's the address.